السلام عليكم ورحمة الله وبركاته. زيدون أحمد جوهر، كلية طب نينا، والمرحلة الثالثة. اليوم عندنا موضوع البكتيريولوجي سيبتوكوكاي بارت 1. بسم الله الرحمن الرحيم، نبدأ الموضوع. Now, first, Streptococci. Streptococci bacteria is gram-positive and negative. They are widely distributed in nature. They are found in normal flora to the human and also may cause diseases in some kinds of these bacteria. Now, classification of Streptococcus is mainly can be classified according to the morphology and hemolysis, the morphology of the bacteria, the shape of the bacteria on the slide or in culture, and the hemolysis. We'll talk about this later. And the basis of cell wall carbohydrate, which resemble the serological tests. And also we have the basis of M protein, which is also for the serology test, and the biochemical tests, and the basis of ecological feature, which is mainly resemble the environmental. First, we will talk about the hemolysis, the Streptococcus. The Streptococcus can be divided into three components according to the hemolytic ability. Now, first group is the alpha hemolytic, which can cause a green color hemolysis around it and in the hemolytic area. This is mainly pneumonia and viridian. Pneumonia can cause pneumonia. We will talk about this disease later. We know about this again, many things. Now, the other type of the hemolytic, which is the beta hemolytic. The hemolytic sample will be clear, complete hemolysis with no green, no any color, just the hemolytic, just bubble around it. There are normal RBCs. Now this caused by pyogenes and agalacia, group A and group B. Now the other type of hemolytic, which is non-hemolysis, gamma group, which is Enterococcus. Just remember this because this is so important, the Streptococcus grouping. The alpha hemolytic causes a green hemolysis sample, partial hemolysis but it's green, partial hemolysis but green. The beta hemolysis caused complete hemolysis and it's clear without any color, just clear color, no color. Okay. Gamma hemolytic, there is no hemolysis in here. Bismillah. Now we will talk about the groups of Staphylococcus types. There is many tests to group the Staphylococcus according to these groups, like A and B and D group. We'll talk about it, but it's not important. Just I have to say it, Now, first, the Lancified classification. The Lancified is a scientist that classified the Streptococcus according to two groups, one from A to H and other from K to U. Now, according to the carbohydrate in the cell wall, that's we talk about the M group or what we call it. Okay, how we do this test? Just extraction from the centrifuged culture treated with a hot HCl and nitrous acid, formamide by an enzyme like pepsin and trypsin, and autoclaving for cell suspensions. And the main groups are important here, which is the A, B, C, F, and G group, which are cause disease to the human. Now, the second type, in what we call it the serology test, other than Lancified, the capsular antigen, the polysaccharide on the capsule of the bacteria. Now, this classifies Streptococcus pneumonia into 90 type. Classify the Streptococcus pneumonia into 90 type, and the other for Streptococcus agalacia group B. Now, the biochemical test will test the reaction with the chemical substances or chemical agents and the presence of enzymes in the Streptococcus, and also the test for the sugar fermentation. The biochemical test is used when the Streptococcus is not grouped under the A, B, C, A, B, F, and G. Sorry, D, F, and G. Now, according to all these tests, all the blah blah blah, I've talked about this just right now. The Streptococcus divided into three groups. The A group A which are the Streptococcus pyogen. Now just remember, pyo mean pus. Pyo mean pus. Streptococcus pyogenes, and B, group B, which is Streptococcus agalacea, and group D, which are Streptococcus enterococci. Now, the three groups are characterized by special morphological appearances, special hemolysis, ability, composition group like cell wall, M groups, and the biochemical reactions. They are all different in all these three groups. Now, we have some examples about this bacteria. Now, not all these examples are mentioned in the lecture of Dr. Shehab, but I will mention the main point and the things that he has said in the lecture. Streptococcus pyogen, I will talk about this later. Streptococcus agalacea, with this is a B hemolysis, beta hemolysis. That's also beta hemolysis. It's called complete hemolysis. This is from group B, B agalacea. This is habiting the female genital tract, cause neonatal sepsis and meningitis. Neonatal sepsis mean التعفن الولادي، تسمم الدم الولادي. Now, Streptococcus dysglacia, it did not mention anything about this in the lecture, but we will study it for interesting. Equisimilis groups and others. This is caused beta, a human infection, alpha, or non. This is found in the throat, causes pharyngitis, pyogenic infections similar to Streptococcus pyogenes. Now, Streptococcus facialis or facalis, or call it, okay. This is a D group. This is called non or alpha hemolysis. This is found in the colon, cause abdominal abscesses, urinary tract infection, and endocarditis. Streptococcus angionosus group, which is for also alpha, beta, and non, found in the throat and female genital tract, cause pyogenic infection. Pyogenic infection is infection with a pus and include brain abscesses. This is so dangerous. Okay. Viridians streptococci, many species. This is cause an alpha. Sorry about this. This is cause alpha or non-hemolytic. This is found in the mouth, throat, colon, and female genital tract. This cause dental caries, S. motus, and which is the S. motus cause dental caries, the endocarditis, abscesses, with many other bacterial species. Now, Streptococcus pneumonia, an important species actually here in this group of Streptococcus, which cause alpha hemolysis, found in the throat, and cause pneumonia, the important disease, the meningitis and endocarditis. Streptococcus, okay. We will talk about the Streptococcus pyogenes, the most important type, which is mentioned in the lecture. Streptococcus pyogenes. This is a slide of Streptococcus pyogenes. Now, first, they are mainly human pathogen. They are PYR positive. So, one of you asked, what's PYR? The PYR is hemolysis of L. pyrrolidonyl 2 nephthylamide. L. pyrrolidonyl 2 nephthylamide. Actually, I don't know what does this mean. Okay. Susceptible to bacterium. This is a spherical or ovoid shape. This is arrayed in chain, like you see here on the slide, arranged in chain, like, and they are produce a 1 centimeter in the zone of culture. They are gram positive and can be gram negative. Like you see, this slide is quite clear, seeing that gram positive and negative, both of them are here, the positive and negative. This produced capsules, young culture. This has a special hair-like projection. They are an M protein. Actually, I have searched many times on internet and Google and Yahoo. I could not find a bacteria with the hair-like. There were no slide in this. I'm so sorry. So just you can imagine it in this. The culture of this bacteria can be in 37 degree. The best media is the human degree temperature is 37. This causes hemolysis will increased with incubation with CO2 gas, and the biogenous strains give two groups. This is important, please. give me attention. Two groups, the mat colonies and the glossy colonies. Now, the mat colonies produce much M proteins that will cause us the virulence, the very dangerous one. Now, the glossy colonies produce less M proteins, and that will cause it not virulent bacteria. So it could not cause that infections or that dangerous diseases. Okay, the antigenic structures. Like I told you, the M protein is very dangerous and would cause the bacteria to be virulent. Now, the major virulence factor of the bacteria is this. It is just like a hair projection, a hair-like shape, hair-like bit. Now, through the M protein, you will not believe this. There is 150 type of this protein. Now, the M protein, there's a special thing about this thing, cause rheumatic fever. Now just remember, or if you have pharyngitis caused by pyogene, or the pyogenous infection, you will get the pyogenes out of your body, but the M protein will be inside your body. Your body will form an antibodies against this M protein. Now this M protein resemble the myocardial muscle tissue, so that will cause rheumatic fever is caused by the attack of the antibodies, your own antibodies to the myocardial muscle tissue. This is the rheumatic fever. I hope you've got benefit of this lecture. Thank you so much, and see you in the next tutorials. Goodbye.